Joining us today to kick off our first What's Next interview is Melinda Kliegman, Ph.D., Director of Public Impact at the Innovative Genomics Institute. Welcome, Melinda. Our topic today is on CRISPR technology. Hi. Can you share a little bit about your background with us? And really, we'll get right into some questions about CRISPR technology. But the first one is, what really is it? Trying to help people get up to speed on what CRISPR technology is and how it's involved in our industries.
Yeah, sure. So I currently work at the Innovative Genomics Institute, which was founded by Jennifer Doudna, who just last year won the Nobel Prize for developing CRISPR-Cas genome editing. And the institute that I work on is a collaboration of researchers from University of California, Berkeley and University of California, San Francisco. And I'm happy to talk to you today about genome editing and some new and exciting ways that it's impacting the agricultural industry.
Thank you. Yeah, one of the things that we wanted to ask is really about the advancements of genome editing in agriculture. And one area is spry technology, which we understand expands the targeting scope of CRISPR-based tools in plant genome editing. Can you tell us a little bit about that spry technology and how it could impact the agriculture industry?
Yeah, sure. So maybe I should just back up a little bit and talk about, I'll give a brief overview about what genome editing is. So it's a group of techniques that allows scientists to very effectively edit the genomes of living organisms. And so that includes microbes, plants, and humans. And one of the most popular types of genome editing is the CRISPR-Cas system. And so that system has been modified from bacteria who use it as an adaptive immune system to cut the DNA of invading viruses. And so researchers have taken that and they re-engineered it to use in other organisms. So in humans, it can be used to correct disease-causing mutations. So for instance, sickle cell disease is one of the first targets. It can also be used as a tool in research and detection. So there is a CRISPR-Cas-based test detection for COVID-19. And then in agriculture, it can be used to protect animals and plants from diseases, increase yields, change nutritional value, potentially also reduce the need for fertilizers and chemical pesticides. So it's really a very versatile and powerful system. With the spry technology, a lot of times you'll hear that CRISPR-Cas allows you to edit any DNA. But that's not really the case. It only allows you to edit DNA that is downstream from a region known as the protospacer adjacent motif, or the PAM. And that is part of the system that CRISPR-Cas uses to recognize what and where to cut. And so that limits researchers to about 1 in 16 sites in the genome. But with this new spry system, in addition to a lot of other Cas variants taken from different organisms that researchers have been finding a lot, it's a very active space of research, you can really now target any region in the DNA. And so it allows researchers to engineer these PAM recognition sites. It's a little complicated how that works, but if anyone wants to learn more about the PAM, there's a really great video on the Innovative Genomics Institute's website, and it's also on YouTube.
Okay, great. Maybe we'll provide a link to that with our article that comes out of this.
Mm-hmm.
That's wonderful to understand at a little deeper level of how this CRISPR technology can help us to mitigate some of the diseases that we encounter in agriculture. What sort of regulations have been put in place in the United States for these products?
Regulators have taken a very practical approach here. So I think it is somewhat informed by the 35 years of experience that they've had with transgenics or GMO plants. With transgenics, a virus or bacteria vector is used to deliver the genes and make the intended changes. And so that typically leaves behind some viral or bacterial DNA. And that's where that term transgenic comes from. And with genome editing techniques, that's not necessarily the case. So you can make a genome edit that is a single base pair nucleotide change in millions of base pairs within any one organism. So these small point mutations that are very targeted can happen really frequently in nature. This happens all the time. And you could also make a change that mimics something that occurs in the organism's natural gene pool. So for instance, you have two varieties of apples. One has been bred for generations to be crispy and delicious, but it's susceptible to a disease. And a wild relative that's small and mealy and doesn't taste so good, but it has disease resistance. So you could just take that one resistant gene and put it into the crispy, delicious apple. And you can do that in one generation. So you don't have to go through the breeding process over multiple generations, especially in things like fruit trees that take a really long time to mature. That process can take a while. So regulators are basically saying, if this mimics something that could have occurred naturally, or that you could do with traditional breeding, then they're just not going to regulate these products. And so in 2020, the USDA published the SECURE rule, and that codifies in the regulations a policy statement to the same effect that was made in 2018. So to answer your question really succinctly, most genome edited products will not be regulated as GMOs. They will just be treated as a conventionally bred, variety of any product.
That's interesting. Can you share with us a little bit about what that looks like from a regulation standpoint internationally?
Yeah, so many countries globally, so most of South America, some Central America, Canada, a few countries in Africa and Asia have all made really similar determinations. Europe was an outlier for a while. The way that their regulations were written, they were treating genome edited products as GMOs. But just last month, they published a study that says that their current law is not fit for purpose. And they are probably going to be making changes relatively soon that are going to be more in line with the rest of the world. And the rationale that they gave was that they see genome editing as a tool to help them meet their aggressive climate targets. And I think there was another big issue with that Europe was facing, is that because these products are so identical to the ones that are found in nature, there's just no way to actually screen for them. So remember, I mentioned those little bits of bacteria or viral signature in GMOs. The European regulators could search the genome for that signature and say, yes, this is a transgenic and a GMO. They could find someone that's trying to import an unapproved GMO into their country. But with the genome edited products, there's no way to tell them apart from a conventionally bred product. So they just had no way of enforcing their regulations. And on a more basic level, if you can't tell the difference between two products, then should you really be regulating them differently? And I would say the answer there is no, that's not really the point of regulation.
It looks like it's heading in the right direction then. Just going back to the crop example, it makes sense that if we can mitigate disease, or like you said with the example of the apples, focus in more on the crispier apples to really cut back the prevalence of the diseases. The question that I think we were wondering about is really how that actually works to help farmers reduce their carbon footprint. Can you tell us a little bit about that?
Mhm. Yeah, so, carbon and nitrogen, which are two strong greenhouse gases, they are both cycled naturally through plants. And so plants, as they undergo photosynthesis, they capture and incorporate carbon by taking CO2 out of the atmosphere. And then a significant amount of carbon is also sequestered in the soil. And this is a function of the interaction between the plant and microbes in the soil, and also microbial communities interacting with each other in the soil. So if you think about decomposing organic material, for instance, some of that carbon is released back into the atmosphere. So you could potentially look into the soil using those genomics tools or machine learning or genome editing, and you can understand which microorganisms in there naturally sequester more carbon while putting more nutrients and organic biomass into the soil. And so you can use these new tools all along the pipeline. So you could edit crops to have improved photosynthetic capacity. So that means they would take more CO2 from the atmosphere. And that means they might actually grow larger and have increased yields. You could edit them so that they exude different types of compounds in their roots, and that would stimulate certain soil microorganisms that would further sequester carbon. You could also edit plants so they have longer and deeper roots. And so the further down in the soil those roots go, the further down they can sequester carbon, and the further down it is, the longer it's going to take to be emitted. So it'll stay stored longer. And so we can also envision that as we go in and do research and learn more about these microorganisms, we can promote certain communities that are more likely to sequester carbon. And all of that means that you have a higher amount of organic matter in the soil. And so the soil is more fertile, and that's going to increase yields, it's going to improve soil health, and it's also going to leave more carbon right there. And so at my institute, we have world leaders in genomics, microbial biology, and plant transformation that can explore all these different mechanisms for using genome editing to sequester more carbon.
Oh, that sounds like a wonderful, really a collaboration to solve a big challenge that we've had. Who would cover the cost?
Yeah, it's really great because you...
Go ahead.
Oh, I was going to say that it's great to look at the system holistically, right? Because if you just look at one component of it, you're missing the other components. It is a cycle. So it's important to collaborate across different types of researchers.
Yeah, that's really awesome. One of the things that we all talk about is climate change, and we want to understand the link that CRISPR technology or genome editing can have on the positive, effects of climate change, like helping that, and help us understand who could take that on, who's going to be responsible for or could own that kind of approach.
Yeah, I mean, my guess is what we're seeing right now is that, because there is such a low regulatory burden, it means that it makes it a lot cheaper and easier to develop these new products. And so I think we're going to see a lot of academic researchers, small companies, developing different types of products and kind of putting them out there. I think the question is then what is the incentive for farmers to use these products? Hopefully it will also be, increased yields, more sustainable practices, better soil fertility. But you could potentially also look to subsidies to help farmers, get paid for farming carbon. So that's also potentially in our future.
I think that is very exciting for the farmer audience. Kind of going back to one thing you mentioned earlier, and that's for animal production. Can you share with us some of the latest advancements as it relates to livestock production, and namely, the food animal production systems?
There are a few genome edited animals in development. So there is a pig that's resistant to PRRS, which stands for porcupine reproductive and respiratory syndrome. And then there's cattle that don't need to go through the painful dehorning process. There are also pigs and cows that have more lean meat. And then for low and middle income countries, we've seen research that's happening around bringing more productive genes from dairy cattle into traditional breeds in Africa that are, for instance, more resistant to heat or more disease resistant. So there's just a lot of activity happening in the livestock space.
Well, I think that could really impact how we raise animals in the future, if we could kind of take some of those elements out of nature that are just cause challenges, whether it's disease or weather resistance or what have you. So that's exciting. Do you see that happening in the near term, or how do you envision what's happening in livestock to become reality for the livestock farmers?
Yeah, I think the technology is there or close to being there. I think the issue with the regulations is more of an issue on the animal side. So a lot of countries just haven't said anything at all. And so we don't really know what their plan is, or they focus on plants. The FDA is the one in the US regulating these animals, and they have said that they're going to treat genome edited animals as a new animal drug, meaning that they view every introduced edit as a veterinary drug that alters the structure and the function of the animal. So I think that that's going to mean long-term oversight by the FDA. So that extends to any offspring that animal might have for multiple generations. And I don't think that approach is really consistent with production agriculture and the breeding process, where one genome edit, may be bred into multiple breeds with hundreds of offspring. And so then that starts to get very, difficult to manage, if not impossible. So I think it's going to take FDA some time, or the US government as a whole, some time to figure out like what the sweet spot might be there for oversight and regulation. And I think there's also, there's this ethical factor or this ick factor. I think because animals are closer to humans in the tree of life and they're prescient, I think that there's going to be a little bit more scrutiny here because people are worried about their well-being. So I think that things will probably go more slowly on the animal side. I actually expect to see widespread genome editing clinical trials in humans with diseases before I think we're going to see animals on the market.
Wow, that's good to know. Don't get too excited then. What kind of?
No, yes, it's going to take a while.
Okay, I think now it's time to really talk about your crystal ball and what you see across all of agriculture and the food system, where the technology is heading. What does it look like 5 or 10 years from now? Kind of share with us from your point of view, how do you see it transforming us, or it helping in our production systems?
Yeah, I think it's really just a tool in the plant breeding evolution and plant breeding pipeline. So I think it's just going to act as a tool to kind of speed up the breeding process and to make new varieties available to farmers faster. I think it also allows scientists to be more responsive to environmental conditions, and hopefully to provide a wider range of options to farmers. Like I mentioned before, we saw a lot of consolidation in the transgenic or GMO industry, where you only have like a few crops. And this is largely because of the cost of regulation. So given that there are fewer expensive regulatory hurdles here, I think we're just going to see a proliferation of different types of products. And I think that's actually really useful to the argument that's happening in the public and the public debate and perception, is that if you have a lot of smaller players, you have a lot of different products, probably a significant chunk of them are going to be focused on sustainability, then I think that this improves the public perception, and that helps, it's like a reinforcing wheel that's going around. So I think it's going to be extremely useful in the next few years, especially as we contend with climate change and a rising population. And then there's also this potential for a new carbon economy that farmers may be able to tap into.
That is really cool to see that come to fruition. Will be exciting for the industry. Do you have anything else you would like to cover with us today or share with our audience that we haven't talked about? I think we covered a lot of bases, and we're trying to see if there's all things related to CRISPR technology that you would be able to share with us, but did we miss anything along the way?
No, I don't think so. I just think it's a really exciting time, and look out for those new products that are coming down the road. You should be seeing them soon.
That's exciting. Well, thank you so much for joining us today, Melinda. It's been a pleasure getting to know you and hearing more about the technology and your deep, deep experience and knowledge across all aspects of agriculture. That's really exciting. And it's often hard to have that all captured in one expert. So that's wonderful for you to be able to join us.